So good morning. Thank you all for joining. I'm happy to introduce Dr. Hanna Poffenbarger, who has been an assistant professor of soil nutrient management at the University of Kentucky since 2018. Hanna received her bachelor's and master's degrees from the University of Maryland and completed her PhD at Iowa State University in 2017. Actually, fun fact, I was one of Hanna's undergraduate lab assistants during her time at Iowa State. So presently, Hanna studies how carbon and nutrients cycle in soil crop systems and uses this understanding to develop sustainable soil management practices and resilient production systems. So welcome, Hanna. We're excited to hear more about your current research and your future goals.
Cool. Thank you very much. So today I wanted to present a little bit of research looking at understanding and managing root carbon contributions to soil organic matter in cropping systems. And I want to mention as well that Lucas Canaceres, he's a postdoc working in my group. He's joining today, contributed to this presentation, and will as well join for some of the discussions. So I wanted to start with this video that depicts its chlorophyll fluorescence in North and South America over the course of a year. And what you see with the bright pink is areas that are have really high photosynthetic activity. And so what we see in general is that the tropics are really productive in terms of photosynthesis, but during that growing season in the southern hemisphere, we see the corn belt of the United States really light up. So that tells us that we have a hot spot of photosynthetic activity in our croplands. So our cropping systems have been optimized for their genetics, their agronomic practices, soil and water management, fertilizers, all towards this goal of having high productivity for food and fuel and fiber production. But so kind of our goal historically has been to increase productivity for the goals of feeding the world. But now society is kind of presenting us with a new challenge, which is can we store some of that carbon that our crops are so effective at fixing? Can we transfer that some of that to the soil and hold it there long term for storage to kind of mitigate climate change? So cropland represents a large stock of actively managed carbon. So about 11% of Earth's land area is used for crop production. And that 140 petagrams of carbon is equal to about 14 years of fossil fuel combustion at our current rate. So it's a pretty large stock of carbon that we have some control over because we're managing it year in and year out. So there's some opportunities and challenges with cropland carbon storage. It's a challenge because in general, cropland is storing less carbon than natural ecosystems. So this figure shows for 3 different long-term cropping system studies, the loss of soil carbon going from when the land was in native vegetation and then since conversion to cropland. So in general, we see losses of between 25 and 75% of the initial carbon stock getting lost upon conversion to crop production. But there's some opportunities as well because since this is managed, we can adjust our management practices to try to reduce loss associated with conversion to cropland, or at least try to restore some of what may have been historically lost from the cropland through what we can generally call conservation management. This could include things like no-till or cover crops, things that are going to help to maintain or build back some soil organic matter. So in general, our goals with managing soil carbon is to either increase the inputs through things like crop residues or manures or compost, or to decrease the losses mainly through respiration. And so there's a variety of different practices. This is kind of I call it a meta-analysis of meta-analyses, because it's a summary of many different meta-analyses that have been done to evaluate different practices' effects on soil carbon storage as well as N2O emissions. And we see, in general, for example, cover crops and no-till, those are common practices that are recommended for soil carbon storage, and they, in general, are going to store more carbon than they would offset through and increase N2O. So in general, these practices seem to be good for creating a carbon sink. And there's another opportunity that hasn't been studied as much that's kind of the focus of my talk today, and that's enhanced root phenotypes. So far, most of the understanding of the potential of enhanced root phenotypes is based on model simulations and just trying to kind of estimate the bounds of what could be possible if we increased root carbon inputs by a certain amount, or deepened the root distribution by a certain amount. So it's estimated that enhanced root phenotypes could fall somewhere in this range, so like 1 ton of CO2 equivalent per hectare per year. So kind of in the same range, or maybe a little bit more storage than no-till would provide. So the reason why the enhanced root phenotypes have been suggested is because roots have a longer residence time as in the soil than shoots. So if you took the same quantity of root carbon and shoot carbon and added that to the soil, you would get estimated 2 to 5 times as much soil organic carbon where you added the roots than where you added the shoots. And that's because the root and rhizopositive carbon can be protected through association or interaction with the mineral surfaces, and that helps to reduce the rate of carbon mineralization. Also, the roots and mycorrhizae promote aggregate formation during their growth and decay, and so that can also help to protect the root-derived carbon from losses. The roots are more biochemically recalcitrant than the shoots, so they maybe contain lignin and suberin at higher concentrations than the shoot materials, and so it causes them to decompose more slowly. And then a portion of the below ground inputs will be delivered deeper in the soil, and typically the deeper we go, the longer residence time the soil carbon has. So I'm gonna, with that kind of background in mind, kind of take you through some different research projects that were motivated by this interest in root carbon and using roots to increase soil carbon stocks. So the first question was, do cropping systems with greater below ground carbon inputs lead to greater soil organic carbon stocks? This is just a general question, like if roots are so good at building soil carbon, can we design cropping systems with greater root inputs, and that will that actually lead to more carbon stored in the soil? So this is work done at Iowa State University, and this was when I was working in the Castellano and Liebman labs, and I've included here the co-authors on this paper as well. I want to recognize Sarah, who helped with this project while she was an undergraduate student. So this study, basically we sampled 3 long-term cropping system studies in Iowa. Kanawha began in 1954, Nashua in 1979, and Marston in 2002. All three of these studies included a 2-year rotation and a 4-year rotation. And as I'll show in a few slides, those 2 rotations created differences in root carbon inputs. So at Kanawha and Nashua, the 2-year rotation was a corn-soybean system with chisel tillage following the corn. And then in the 4-year system, it was corn, oat interseeded with alfalfa, followed by a full year of alfalfa. And in that system, there was also chisel tillage following corn and following the alfalfa. At Marsden, the systems were similar, except the 4-year system, the second phase there was soybean rather than corn. And it also received some manure inputs in the 4-year system. So I calculated below ground inputs and above ground inputs using the yield data that we had from the years of these studies, as well as the harvest index and root to shoot ratios. So these are estimated inputs based on those allometric relationships. And for the Marsden site, it accounts for the manure inputs as well. So in general, the 2-year and 4-year systems added about the same amount of carbon back to the soil, averaged, for an average year. However, the 4-year system had 20 to 35% more below ground inputs than the 2-year system. So basically the same overall carbon inputs, but a greater below ground allocation in the 4-year system. So we looked at the soil carbon stocks for 0 to 90 centimeters. Those are broken down into different increments. You can see on the y-axis. And then we have the 2-year in red and the 4-year in blue. So at Kanawha, there was a significantly more soil carbon in the 4-year system for the whole 0 to 90 centimeter profile, and also specifically at 0 to 15 centimeters and 60 to 90 centimeters. In Nashua, we saw a similar trend where the 4-year had numerically more soil carbon than the 2-year, but it wasn't significant. And then at Marsden, which is the youngest experiment, we didn't really see any evidence that the 4-year system had more soil carbon than the 2-year. So some evidence for this idea that a rotation with more below ground inputs will lead to more soil carbon, though it was pretty inconsistent among the locations. Wasn't a super strong effect. We also wanted to look at the composition of that organic matter. Specifically, we thought that the greater root inputs of the 4-year system would lead to more plant-like carbon at depth in that rotation. So we looked at two different ways of looking at the plant-like carbon. The first was fractionating the soil to get particulate organic matter versus mineral associated, because the particulate organic matter is more plant fragments, whereas the MAOM is a mix, but more on the microbial derived side. And so this figure shows on the x-axis is pre-palm carbon as a proportion of the total soil carbon. And then we see the different depths on the y-axis, and then the different systems are different colored points. So overall, we did not see an impact of the rotation on the proportion of carbon found as free palm. We did see an interesting depth trend where it kind of decreased and then increased to the deepest depth. And then we thought that may be due to the increase in sand at depth in these profiles. We also looked at the ratio of galactose plus mannose to arabinose plus xylose. So galactose and mannose are 2 microbial-derived sugars, and the arabinose and xylose are plant-derived sugars. So the fact that the ratio was lower at the surface tells us we have more of plant-derived organic matter, or at least plant-derived sugars near the soil surface. And then as you go deeper, you're getting more of that microbial influence. So it seems like the deeper soil is more microbially processed. That we can definitely see the reflection of above ground residue and shallow roots, leading to more plant-derived makeup of the organic matter near the surface. But with this metric, we also did not see any impact of the rotation. So again, we don't see much evidence for those root inputs leading to more plant-like carbon at depth in the Fourier system. Okay, so overall, kind of minimal effect of crop rotation on the profile carbon stocks and composition. But we also, we looked at the soil carbon changes over time in 2 of the long-term studies that had previously been sampled in 2002. So we looked at the change in soil carbon between 2002 and 2014 and calculated the soil organic carbon change as a percentage of the initial by depth. And what we found was that the surface soils showed a loss of soil carbon at the soil surface. And then as you went deeper, there was more of an increase in soil carbon. We were kind of surprised by this trend, this depth effect, and also how consistent it was between the 2 sites and also the 2 rotations. So then we also, we looked at 2 published studies that had done resampling to look at soil carbon changes by depth. And that was the David et al. 2009 and Beanstra et al. 2015. For both of those studies, they were they had resampled soils that had been sampled back in the 1950s. So they looked at changes between the 1950s and the early 2000s. And these were done in Iowa and Illinois. And they found something very similar. When we compiled all that data along with ours, we saw the same trend, that there was a loss of carbon at the surface and an increase at depth. We don't really know why. So that was what kind of led me to the next question. The next question was, how have root carbon inputs changed due to breeding? Because one hypothesis or explanation we had was that perhaps over time, the crop root systems have gotten bigger or they've gotten deeper, and maybe that would explain why there's this increase in subsoil carbon over time. So this is a study that we conducted at University of Kentucky with this group, and this is Brian Reinhardt and Travis Bannett, 2 graduate students, Dave McNear, Monse Salmron, and Charlie Messina. So we did a greenhouse study, and this was using an ERA panel. So it was the pioneer hybrids spanning the 1930s to modern corn hybrids, kind of commercially popular hybrids. And we took 12 of them, so 3 from each of 4 eras, and planted them in the greenhouse in these tall PVC pots. And we grew them for 28 days. So it reached about the V7 stage, and then we washed off the roots and imaged them on this big imaging table that you see in the center photo. With those images, we extracted the root length, the root diameter, root depth distribution, and we also analyzed the tissues for carbon and nitrogen concentrations. So these are just some images to show what the systems looked like at different time points. So the 1946 hybrid and 76 hybrid appear larger, especially compared to the 2014 hybrid. That root system definitely visibly looks smaller. We looked at the total root length for each hybrid. So this is showing on the x-axis the year of release, and the y-axis is the root length, and then their boxes are colored based on the eras. So the first era, the double cross hybrids, had significantly higher root length than the subsequent eras. We also found that the oldest hybrids in the double cross era, they had about 30, I think it was 35% more mass on average than the subsequent eras. We also looked at the root depth distribution using both root length and root mass. We fit curves to the depth distribution to figure out the depth at which 50% of the roots were above or below that depth, and then the maximum depth. So for both the root mass and the root length, the double cross had a deeper D50 and D max than the later eras. So we found in general that the oldest hybrids were also more deeply distributed than the newer eras. So I wanted to kind of compare this to other era panel studies because I wasn't sure if this was just a pattern observed with the Corteva hybrids or if this is something kind of generally happening with breeding. So I compiled some different era studies where they reported root biomass. We see on the left wheat and on the right is corn, and the y-axis is relative root biomass. So these different studies were conducted in different environments, different growth stages. So I've relativized them all based on the most productive root system within each study. But generally what we see is the wheat root systems do show this decline pretty consistently among those different studies, the newer varieties having smaller root systems than the older ones. With corn, it hasn't been as consistent of a pattern. You can see the Reinhardt green line there is definitely showing a decline, but some of the other studies show actually an increase or no change over the breeding history. So it seems like with corn, it's been a little inconsistent in terms of how the root systems have changed with breeding. Okay, so the third question then is, if the root systems, at least for this corn panel, have gotten smaller over time, how is that going to impact, or how has that impacted soil organic carbon levels? So this is the kind of the follow-up study to the greenhouse study. We're taking the two oldest and the two newest hybrids from that greenhouse study and planting them in the field. We're doing old and new plant populations because plant populations have gone up over time. So the modern hybrids are planted at a higher density than the older ones. We've taken a field out of C3 grasses so that we can use natural abundance differences to differentiate the carbon coming from the corn versus the old carbon. And we also have a residue removal treatment. So we can specifically kind of isolate the effects of the roots versus the roots plus above ground residue. And then this study started this last year, and it will continue for a few more years, at least 2 more years, and hopefully longer. With this study, we also have mini rhizotron tubes installed in order to track the root growth, the root length and diameter of the different hybrids during the growing season. So with the mini rhizotron data, so far we've processed the images for the V3 and V7 stages from 2021. This is the high plant population. We see at V3, there's not a lot of roots present yet, so not a whole lot of treatment effects. But at V7, we're starting to see those older hybrids having more root length than the newer ones. And that was significant at that intermediate depth of about 20 to 40 centimeters. So far the field study does seem to align with the greenhouse study, showing like lower root length with the newer hybrids. We also looked at the yield from 2021, and the modern elite group, the two newest ones, definitely had a higher yield than the older ones. But the other thing we noticed is that the modern hybrids demonstrated an advantage at the hive populations. So for the modern hybrids, we definitely had an increase in yield, moving from the 30,000 to 70,000 plants per hectare, whereas the double cross didn't really respond to the population. And so one kind of a working hypothesis that we have is that maybe the smaller root systems of the modern hybrids are one factor that enables the crops to grow better at this high population. Maybe there's less below ground competition between plants with these smaller root systems. So now onto the final question that we'll present on today. How do living roots affect the turnover of soil organic matter? This is research led by Lucas Caniceres and also with collaboration from Travis and Brian and Dave McNear. So we know that roots can efficiently form new soil organic matter, but there's also evidence that they can accelerate the loss of pre-existing organic matter in what we call the priming effect. This creates what's been described as like a double-edged sword. Like it's kind of unclear whether they will be as larger root systems will be as beneficial as we might think if they're having this priming effect. So on the left side, we see some of the ways that roots are building organic matter. They're releasing carbon as they're growing, and then the litter after they die, those root inputs contribute to mineral associated and aggregate protected forms of organic matter. But then with the rhizodeposition and the exudates that they're producing, those may help to stimulate microbial activity, and the microbes could then go for more stable forms of organic matter. And so it's possible that you could get kind of offset, that priming effect could offset some of the benefit of the added carbon. So there's some research to show that this priming effect can be more pronounced under conditions of nitrogen limitation. So to describe this properly, I should first describe a little bit about the two fractions that you see here. So when you talk about the roots priming organic matter that's more stable, we're thinking about the mineral associated organic matter. That's the organic matter that's bound to clay and silt particles. It's typically more microbially processed, slower to turn over, and with a lower C/N ratio. Whereas the particulate organic matter, faster cycling, more plant-like, plant fragments, for example, and a higher C/N ratio. And so when microbes are living in an environment with a low nitrogen supply, they may go for that mineral associated organic matter because it's a lower C/N ratio. So we developed some hypotheses about litter quality, specifically litter stoichiometry, and how it would affect mineral associated organic matter. So moving from left to right with these pots of corn, we have carbon to nitrogen ratio of 20 on the left and then up to 65 on the right. So we're going from a lower C/N to a higher C/N ratio. So as I mentioned, we might expect more of this rhizosphere priming effect in the high C/N ratio, where the microbes are going to be more limited by nitrogen. So they may start to break down that MAOM. However, with this green triangle, I want to point out there's also some evidence to suggest that the low C/N litter could be more efficiently converted into mineral-associated organic matter, because that ratio of the carbon and nitrogen is going to be a better match for what the microbes need to grow. So they'll use the carbon more efficiently rather than kind of respiring extra as they might under a high C/N ratio litter. So with those two processes in mind, that led us to this idea that the low C/N ratio would be our blue ribbon winner in terms of like best MAOM in show. Whereas the high C/N ratio litter, maybe that would have the lowest MAOM due to the combination of less efficient MAOM formation and more impact due to rhizosphere priming. So this is a mesocosm study conducted in the greenhouse. We have on the left side no litter, just soil. And we have soybean, buckwheat, and Sudex residues added. And the C/N ratio for those range from 21 to 65. I'll mention this. All three of these were summer cover crops. So the soybean is a higher quality than what we might expect following harvest. It's not as mature soybean plant as you would find following a harvest. And then we have different plants. So we have oats, which are shown in the front, or no plants. The oats I've drawn in because at the time of this photograph, they weren't growing yet. So those are just illustrated in this case. And we used a soil that had a delta 13C intermediate between the soybean and buckwheat and the Sudex. So it was, I think, around -20 or -21. So we wanted to see how much of the carbon that we measured in the soil at the end of the study was coming from the litter versus the old soil organic matter. So first off, I just wanted to show that the buckwheat and Sudex litters were successful in terms of creating nitrogen stress. We definitely saw much lower nitrogen uptake by the plants in the planted pots with buckwheat and Sudex. And then soybean, we saw better growth and more nitrogen taken up in that treatment than the no litter treatment. Okay, so this figure shows, first off, we're showing with the no plant treatment. On the y-axis here, we have the carbon content, and the different fractions are shown with different colors. On the x-axis, we move from left to right. We have no litter, soybean, buckwheat, and Sudex. So it's going from no litter and then increasing in C/N ratio from left to right. So one thing we noticed in this unplanted treatment is that the Sudex on the right there, it had the greatest litter-derived mineral-associated organic matter. This was after about 40 days of incubation and plant growth in the greenhouse. So that's about how much decomposition had taken place. It was about 40 days worth. So we did find the most litter-derived MAOM in the Sudex, but I want to point out that overall the mineral-associated organic matter, old plus litter-derived, was about the same. So it seems like the Sudex, it added more litter-derived MAOM, but there was more of a loss of the old organic matter as well. So it's kind of seemed more of a replacement of the MAOM than adding on to the pre-existing MAOM. Also want to just highlight that that green bar is representing the undecomposed litter. So the Sudex definitely had the most undecomposed litter remaining. It was decomposing more slowly than the buckwheat and the soybean. And you can see the buckwheat mainly ended up in the palm fraction. Now I've added on the right side here the oat treatment. With the oat treatment, we did find the increased incorporation of soybean and Sudex into the mineral-associated organic matter, and that's what I've highlighted in the yellow outline. So going from soybean without a plant to soybean with the plant, we had more litter-derived MAOM, and then Sudex decomposing without the plant to with the plant, we had an increase in that litter-derived MAOM as well. So it seems like the living plants, perhaps they are kind of stimulating microbial action, and that's helping to transform the litter into the mineral-associated organic matter. We also calculated a retention index, which we defined as the mineral-associated organic matter from the litter relative to the unrecovered litter carbon. So it's saying of what was processed by the microbes, how much is going into the MAOM for each of these litter types. With the oats, we saw more of a pronounced difference among the litters, where the Sudex really had a higher retention than the other two litter types. And that was not as obvious of a litter effect when there was no oats growing. Okay, so returning to those initial hypotheses, we didn't really find a litter effect on the overall amount of MAOM, so they all get to share the blue ribbon. But we did find some interesting things with what was going on with those processes. So with the efficiency of MAOM formation, it actually seems like the Sudex was more efficient at transforming into mineral-associated organic matter, which was the opposite of what we hypothesized. The Sudex had the highest C/N ratio. And we thought perhaps it has to do with the type of microbes that are breaking down that Sudex. Maybe the community that's breaking down the Sudex is more efficient overall than the microbial community that was decomposing the soybeans. But we don't have that the microbial community composition in this study. The other process here was the rhizosphere priming. We didn't find significant effects of the litter type on the loss of old organic matter, but there did seem to be a trend with the Sudex having more of the loss of old organic matter. So overall, it seems like the Sudex was kind of stimulating the turnover of the MAOM more so than the soybean, but there did not seem to be an overall loss or gain in the MAOM as a whole. So kind of as a next step, we thought we learned a lot from this study. We did identify kind of a weakness was that we couldn't differentiate what's coming from the plant, the living plant rhizodeposits versus the litter and the old carbon. We only were able to differentiate the litter carbon and the old carbon. And we did our best estimate of accounting for plant rhizodeposits, but we weren't able to directly measure that. So we had an idea to use actually 13C and 15N labeling of particulate organic matter and mineral-associated organic matter to try to get a better handle on what's going on with each of those components. The plant litter input, the plant carbon inputs, the litter fraction, or the litter/palm, and then the mineral-associated organic matter. We feel like this would be a good step because a lot of priming studies just kind of separate plant-derived from soil, but we feel like getting a better handle on what's going on with each of the soil fractions would help, since those soil fractions have different properties, turnover times, and formation processes. So it's kind of something that we may have time to discuss in the meetings this afternoon, is possible next steps with this line of research. So with that, I just want to say thank you. I got lots of people both at University of Kentucky and Iowa State who've helped with this research that I presented today. And also want to acknowledge the various funding sources. And I think that's all I've got, if we have time for questions.
Great. Thank you, Hannah. That was lots of great information, and I'm sure it'll spark a lot of great conversation later today. So does anyone have any questions? Because I have one to start. This sort of goes back to one of your first questions about looking at how over time we sort of have a decrease in this root biomass below ground. How do you balance both still trying to keep that below ground root biomass, but then also breeding for higher grain production? Is that sort of the trying to balance those two? That's the issue?
I think it could be an issue. I don't know yet for sure, but I do know that there's clearly a correlation where over time yields have gone up and per plant root biomass has gone down. So that we don't know if that's caused, the lower root biomass is causing the higher yields, or that's just a correlation. There's some other relationships between yield and root tissue ratio. So usually, I don't know usually, but I've seen evidence that yields are higher when you have a lower root tissue ratio. So that would be another piece of evidence, thinking there might be a trade-off here between what's going below ground and what's going to the harvested product. But then there's also some benefits, like if you have bigger root system, especially during vegetative growth, the plant can capture more resources, and then it can help to produce more grain later in the season. So I think that there could be a trade-off there, but maybe not necessarily a trade-off. But that's a good question.
Yeah, thanks for that. Looks like Kirsten has a question.
Hi, Hannah. Thanks for the talk. That was really interesting. I was curious about, in thinking about C to N ratios of the litter inputs, and also, in some of your studies, you were using the same soil, so you're actually controlling for any background C to N. But soybeans an N fixer, and so you would have additional N coming in that could affect microbial processing and sort of that efficiency. And so I wondered what your thoughts were on retention and how that additional N input might influence carbon storage and cycling.
So you're kind of referring more to the rotation studies than the, yeah. Yeah, so in the rotation studies, the Kanawha and Nashua sites, they got the same nitrogen rate across both rotations. And we have the soybeans fixing nitrogen, we also have the alfalfa fixing nitrogen. And probably a farmer might not apply as high of a nitrogen rate following alfalfa than soybeans. So I think your question is very good, because I think there was extra nitrogen in the 4-year system.
Yeah.
Because it's getting the same amount of fertilizer but extra alfalfa nitrogen. And so I think I'm not as much of an expert on the microbial side, so I don't know really what that means. Perhaps the microbes are kind of relaxed because they're like, well, we got so much nitrogen, we don't have to break down organic matter as much. So, but maybe they're stimulated because they're like, oh, we've got a lot of nitrogen, so now we can do more work. I would be interested in your thoughts on what that extra nitrogen might mean.
Yeah, it's interesting because with the N fixers, there seems to be this synchronicity with microbial demand and that rise of the carbon inputs as well, right? So you have this like couple of carbon and nitrogen. Whereas, as you know, with like the fertilizer, it's not super efficient. It kind of comes as a big pulse, and it can alter the microbial resource availability relative to like carbon, right? Which would be important for thinking about biomass building versus respiration. I thought it was interesting too with the soybean, when you had the soybean and the Sudex and buckwheat, right? Is soy is the only N fixer in that one as well?
Yeah, but with that one, it was just the litter, so we didn't have soybeans growing.
Oh, you didn't have the plants in there, right? Yeah.
Well, the plant was oats or no oats, so it was just grass.
Yeah, so it could be interesting to compare across those two, because in that one you actually, in some ways, control for that N fixing part.
That's a great point. Yeah.
Yeah. Cool.
Cool. Thanks.
Looks like Eric has a question. You want to go ahead, Eric?
Yeah, in both the field studies that you're doing, and also I think you looked at some the historical data. Were these strictly tilled fields, and does no-till change the this sort of effect? Because with tillage, you're turning under all the litter, whereas with no-till, I think the roots would be a major the only major source that's deep.
That's a good point. For the long-term crop rotation studies, those were all tilled. For the published studies that I showed that also kind of showed this increase in deep soil carbon, those were what I would call observational. So they weren't controlled. They were just a field that was sampled historically and a field that was sampled again. So it kind of reflects general practice. It's possible that some of those fields went from tilled to no-tilled over that period, but we don't know for sure. So it was probably just a mix of different management practices. And then the field study with the corn hybrids in Kentucky, that was plowed to convert it into from the grassland into corn, but then we're maintaining it as no-till because we do want to best capture that root distribution without kind of mixing things up. Because I think your point is good that that tillage process is going to introduce above ground residue deeper down, and in that study we really wanted to look at just how the root distribution is affecting the distribution of soil carbon.
Yeah, I just sort of noticed that some of your data showed fairly linear trends with depths, and then I think somebody else's data, they plotted a curve on it, and it the sort of apex of that curve I think was around 20 centimeters or so, which is about the depth of a plow. And so I'm wondering if that's is going to be sort of showing a difference between till and no-till, at least these shallow depths.
Yeah, I think that's a good point. Yeah.
Do you want to go ahead to Andy? It looks like you have a question.
Yeah, I was just curious on the three, what is it, Iowa sites that you showed earlier. What's the difference in like the mineralogy in those soils? I mean, since they are so relatively close together, I mean, are there any real differences?
So yeah, let me go back to the Marsden and Kanawha were both on what we call like the Des Moines lobe, which is a more younger soil, glacial till derived, high clay, kind of naturally poorly drained. Whereas the Nashua one, see this brown portion of the map, that's the lobe. And then the Nashua was off of that, and that was more of a weathered, older soil. more red in color, lower in organic matter. So I don't know the mineralogy, but I know that the Nashua was a little different than Kanawha and Mars, and those two were more similar.
Okay, thanks.
Erin, do you want to go ahead? You have a question too?
Yes, I have a question about microbial association with the different hybrids over time. So like you can see the difference in root structures, and what I was wondering was whether you looked at microbial communities that are associated with hybrids and whether it's known whether the different varieties that you were able to grow from different historical periods had any differences as far as fungi or bacteria that were associated.
Yeah, we haven't looked at that. I think there have been a few studies along those lines, but I'm not sure that there has been a consistent trajectory in terms of like in general breeding is leading in one direction with that. I know I've reviewed some papers on like the mycorrhizal association, and I think that was also a little mixed if I remember right. There's kind of this idea that the modern hybrids bred under high nutrient conditions would have less of a relationship, or maybe a weaker relationship with those symbionts. And especially, I think if you look really far back to like the land races, that maybe that's evident, some loss of that symbiosis, but then within more of the modern panel, like from the 1900s to now or something along those lines, that the change in the symbiosis isn't as strong. That's what I remember from reading some on this topic, but I haven't looked at it myself. But I know that there is this idea that that the breeding process in really high resource environments may affect, you know, what microbes the plants are associating with below ground.
Thank you. Yeah, it would be fascinating to see if there are genetic qualities in the plants that are affecting like that association, and see if that could be accounted for in future breeding practices. I'm intrigued. Thank you.
Yes. Thank you.
Those are all great questions. Anybody else have a question? We have a few more minutes left. Yeah, I guess going off that last question, do you have any thoughts, Hannah, on any sort of microbial tactics you might incorporate in future studies, or anything along those lines?
Yeah, so especially in that last mesocosm study, we were really left with this question about the microbes that were present in those different treatments, and if the microbial community composition would help to explain the MAOM formation that we saw with the Sudex. So that, I think that's something that we want to incorporate in the next phase that I mentioned right at the end, in terms of trying to make this study a little bit more mechanistic. So I don't have as much expertise on the microbial side, but I think Lucas does, and so he's got some ideas about measuring maybe PLFA or other measurements that we could do to get a better handle on which type of microbes are there.
Yeah, really exciting work. Well, I don't see any more questions. We were very efficient with getting those questions answered early, I guess, which is great.
Something maybe in the conversation. Let's see. I think, yep. Well, thank you all very much.
Yeah, great talk, Hanna. Thank you. Great. And yeah, the rest of you'll get a chance to talk to some EMSL scientists later today and kind of ask them about how we could potentially use some of the EMSL instruments to help you answer some of your research questions. Great. Well, thank you everyone for attending.